Self-fractionation. So for the better understanding of a living organisms, we need to study the humans at a very small level, which is obviously a cell level or at organelle level. So we have to study the anatomy, which is the internal structure of a cell, and the physiology, which is the function of the organelles and the cell. So we need to study the anatomy and the physiology of a cell. So for that, there are a lot of techniques which have been applied at the laboratory level. One of the technique is known as a self-fractionation. If we look into it, the animal cell and the plant cells, its size varies from a 10 micrometer to 100 micrometer. So the organelles which are obviously present inside the animal cell and the plant cell, their size is smaller than the 10 micrometer. For example, if we look into this mitochondria, its size is 1 micrometer. If we look into this proteins, its size is 10 nanometer. So all the organelle, they have a specific structure and they have a specific size. So on the basis of a size and the densities of these organelles and the cells, self-fractionation has a done. Why self-fractionation has done? Because it helps us in the studying of these structure and the function of these organelles through electron microscope. So self fractionation actually takes place in two steps. The very first step is known as homogenization, and the second step is known as differential centrifugation. So what we do, we take the tissue cells or tissue cells of an animal cell or a plant cell. Firstly, we homogenize them. After the homogenization, we will perform a differential centrifugation on this homogenate. Homogenization, which is the very first step of a self-fractionation. If we look into this word, homo means same and genization comes from the word genicity, so same composition. So we need to create a cell homogenate having a same composition. We need to perform a cell homogenization? The very first purpose is that we need to study the cell organelles, and to study them we need out of the cell wall or the cell membrane. Cell wall is obviously outer covering of a plant cell and cell membrane is the outer covering of all the cells. And in case of plants, we have an external membrane which is known as a middle lamella, which connects the one plant cells to the another. So to digest the middle lamella, we also have to add the enzyme pectinase in case of a tissue cells of a plant cells. But in case of a tissue cells of an animal cells, we don't have to add enzyme pectinase, because in tissues the cells are connected with each other through the cytoskeleton, not with the middle lamella. So what we do, we take the tissue cells, we grind them under a specific conditions, which is the pH, ionic composition and the temperature. Why we have to maintain these three conditions? Because we know that inside the cells there are a lot of enzymes, there are a lot of the functional units, and their functions get disrupted if the temperature or the pH or the ionic composition changes. So under a specific condition, which is pH, ionic composition and the temperature, we will grind these cells and we will create a cell homogenate having the same composition. Centrifugation, which is the second step of a self-fractionation. So what we are supposed to do in centrifugation, हम लोगों ने first step में क्या किया था? Homogenization में we have created a cell suspension. We will take that cell suspension, cell homogenate, and put them into a centrifuge tube, and then this centrifuge tube is gonna be placed in this centrifugation machine. For example, if we are placing this centrifuge tube at this place, so we need to put the same test tube of a same size with the water. If for example we don't have any cell homogenate at the opposite poles, हमें इस चीज़ का ध्यान रखना पड़ता है कि जब भी हम सेंट्रीफ्यूगेशन को परफॉर्म करते हैं तो जो हम सेल होमोजिनेट लेते हैं, जितनी अमाउंट में, जितनी क्वांटिटी में सेल होमोजिनेट हम ले रहे हैं, सेम ही क्वांटिटी में अगर हमारे पास कोई और सेल होमोजिनेट नहीं है एट द सेम टाइम, सेम ही क्वांटिटी का वाटर आपने लेकर किसी और सेपरेट ट्यूब में डाल के अगर आपने यहाँ पर सेल होमोजिनेट रखा है तो उसके ऑपोजिट पोल्स पे आपको यहाँ पर वाटर डालना पड़ता है टू क्रिएट अ बैलेंस. व्हाई? बिकॉज इन अ सेंट्रीफ्यूगेशन व्हाट वी do, we rotate this rotator at a very high speed, and because of that rotation it creates a centrifugal force, and that centrifugal force separates the cell content or the cell organelles on the basis of their size and densities. So जो इसकी rotation होती है that is very very high, उसकी speed बहुत ज़्यादा होती है. जो minimum एक centrifugation machine की जो speed है that is 300 rpm. that means in a single minute 300 times this rotator is going to rotate, और जो maximum होती है it is like 150,000. So minimum be 300 है, in a single minute it is gonna rotate 300. So because of a 300 times rotations, if there is nothing at the opposite pole, so balance नहीं create होगा and cells homogenate is gonna spill out of this tube. So we need to maintain a balanced. So what we do, we will simply place the cell homogenate over there, and as another tube with the, for example, water at this, close the lid and switch on the centrifugation machine at a specific rpm. This rotator is gonna rotate at a very high speed, and that speed of rotation will create a centrifugal force, and because of that centrifugal force, all the organelles of a cell are gonna be separated on the basis of their sizes and the densities. Centrifugation has basically two types. The very first type of a centrifugation is known as a differential centrifugation. Why we call this centrifugation as a differential centrifugation? Because in differential centrifugation, the cell organelles are gonna be separated in a series. There will be a different sets of these centrifuge tubes. That's why we call it as a differential centrifugation. What we do, we take the homogeneate cell homogeneate, and at a very low speed, at a very low rpm, we will spin it, we will rotate the centrifuge machine, and in a first step, centrifuge is gonna separate all those organelles having a high weight, having a high molecular weight. For example, nucleus and cytoskeleton usually have a very large size. So because of their large size, they are gonna be settled down at the bottom, and we call this as a pellet, and the above cell suspension is known as a supernatant. What we gonna do, we will take the supernatant and then we centrifuge it at a higher speed as compared to the speed that we took the first time. So हमने इस टाइम में ज़्यादा स्पीड लेनी है, ज़्यादा आरपीएम्स लेने हैं. At this time, those organelles जिनका size smaller होगा as compared to nuclei and the cytoskeleton, but it will be a larger than the other organelles that are gonna be settled down at the bottom, and we will call it a pellet. और जो supernatant होगा, we will take it into a next tube and at a higher speed than the previous two ones, उनको हम दोबारा से centrifuge करेंगे और एक pellet create करेंगे, और जो जिनका smallest size होगा इनसे भी that will be centrifuged at the last. So what we do, we performed a series of a centrifugation, and in each step different organelles of different sizes and the densities, they will be settled at the bottom, known as a pellet, and the smaller size will be present on the upper test tube, upper part of the test tube. We call it as a supernatant, which will be separated on the next centrifugation. Density gradient centrifugation, which is a second type of a centrifugation. In a density gradient centrifugation, we don't have to perform a series of a centrifugation. In a single centrifugation, the cell homogenate components on the basis of their size and the densities, they will be separated in a single test tube. We don't have to perform a centrifugation multiple times. In a single centrifugation, all the components are gonna be settled down in a single test tube according to their size and their densities, and there will be no supernatant and the pellet in case of a density gradient centrifugation.